Hi, let's talk a little bit about ribosomes. Let's do a little deeper dive into it. So these are organelles that read your RNA and create proteins in both prokaryotic and eukaryotic cells. So they're one of our very ancient old organelles in that they are in both prokaryotic and eukaryotic. So that's how we know as scientists that they've been around for a very long time. So here we have just a real basic generic animal cell, but remember ribosomes are present in both eukaryotic and prokaryotic. So they'll be in plant cells, they'll be in animal cells, they'll be in bacteria cells. The only cell type they won't be in is none because they're in both cell types. So we have our nucleolus, our nucleus, our endoplasmic reticulum, our beautiful mitochondria. Those are very important, our powerhouse of the cell. We have vacuoles, because more than one, because this is an animal cell, not a plant cell. Lysosomes, those show up. We got all sorts of that jelly-like cytoplasm. We have a nuclear membrane around our nucleus. And last but not least, our beautiful ribosomes. And your cells are going to have more than one ribosome. It's not like a nucleus. There's going to be many present. So let's break that word down a little bit. So ribo references the carbohydrate ribose, which is the structural backbone of the ribosome. And some, or soma, is a Greek word, and it just means body. So ribosomes are organelles inside prokaryotic and eukaryotic cells, and they read your RNA and create proteins. So ribosomes are responsible for building proteins, which are those essential biomolecules that make up your enzymes, your hormones, and even your cell structures. And ribosomes follow all of their instructions from the RNA, which is made using DNA's code. One of the cool things about ribosomes, other than the coolest part being that they're in both prokaryotic and eukaryotic, it means that the bacteria inside your body has its own ribosomes, and your own cells, your body cells, your animal cells in your body also contain unique ribosomes. And they're all working inside the cells, the prokaryotic bacterial cells and the eukaryotic animal cells of your body to survive and grow. There are some, so I mentioned bacteria cells, those prokaryotic bacteria cells could maybe be infecting you with something that's making you sick. Well, those bacteria cells also can be targeted. There's two antibiotics that are commonly prescribed that focus on ribosomes within those bacteria cells. So if you've ever taken antibiotics like tetracycline or streptomycin, those work by targeting specifically the bacterial ribosomes, which are different enough from your own animal cell ribosomes that you can attack them and not harm your human cells. So that's one of those really neat things that we found as scientists. So we already talked about how ribosomes are found in both eukaryotic and prokaryotic cells. Well, they're doing the same type of job within both cell types. There is a size difference. In prokaryotic cells, ribosomes tend to be significantly smaller, just that's very common. In eukaryotic cells, ribosomes are bigger. But they're doing the same type of jobs in both cell types. So they're creating proteins, protein synthesis, they're reading RNA, and they're also linking amino acids, which that is going to be something that you would cover more in detail in probably a college biology class, not in our current class. But those are the things that they're doing, and they're doing them in both prokaryotic and eukaryotic cells. Now, here's where it gets interesting. Remember, viruses do not have ribosomes, but they need ribosomes. How they get access to ribosomes is how viruses get access to any metabolic feature or including reproduction. They have to hijack a host cell. So viruses are not living, and this is one of the reasons. They can't make proteins by themselves. They have to hijack a host cell in order to do all of those metabolic features and reproduction. So by hijacking a host cell, viruses can trick the host cell into making those viral proteins instead. So they're using the ribosomes to make those viral proteins. So because the virus lacks its own ribosomes and they can't make them without that hijacked host cell, the host cell ends up being injected with that virus RNA or DNA, because viruses contain either DNA or RNA. And that hijacking process, the virus is tricking the host cell into using its own ribosomes to make the viral proteins. So that's how they're reproducing in a way. The virus hijacks your own protein making machinery. So enzymes and ribosomes. So stepping a little bit away from viruses and looking instead back at maybe bacteria and infections, bacterial infections. So ribosomes are responsible for making proteins, including enzymes. So that's important. But I want you to remember we talked about two specific antibiotics and the target ribosomes. So because bacteria depend on enzymes to digest nutrients, build their cell walls, reproduce, and carry out all metabolic features, enzymes and ribosomes are very unique and they're connected. So in a normal functioning, without antibiotics, you can think of some of that healthy bacteria that's in your digestive system as having that. Remember, the enzyme would be the lock. So that would be your bacterial ribosome. That would be the lock. And then the key would be the RNA that's bringing those amino acids to build the proteins inside, including enzymes. So the result would be, once you introduce your lock to your key, proper protein enzymes are going to be made. So in healthy bacteria, the ribosome is working, it's like a working lock. It's the RNA key fits perfectly and turns, and it allows the bacteria to make the enzymes and survive. However, there are those two specific antibiotics that we work, and they're not destroying enzymes, but they're stopping the new ones from being made. So these two specific drugs, tetracycline and streptomycin, they work by focusing on stopping the enzyme production, which means they are targeting the ribosomes in those bacteria. So with tetracycline, that's sort of like putting gum inside the lock. So it's blocking the active site on the ribosome. So you can't get the key inside the lock. Remember, we use that lock and key metaphor? So this is one way that we have this tetracycline is blocking the active site. So your enzyme and your substrate cannot interact. So that's how you prevent new enzymes being made. There's no ability for that ribosome to make more protein. So that's just a really neat example. Whereas with streptomycin, it's not blocking the active site. This actually, as an antibiotic, damages the key. It impacts the substrate. So it's like bending the key or maybe shaving off one of the teeth on a key. It's not the same shape anymore. You've changed or damaged the key shape. So the substrate isn't going to fit inside the lock as well. So it can't make more proteins. It can't recreate more. So you're not destroying the enzyme. You're not ruining the lock. But with tetracycline, you're preventing access to the active site. And with streptomycin, you're damaging the substrate. You're preventing the substrate, the key, from being able to fit inside that lock anymore. So it ends up making the wrong protein or not making any protein. So this is one way that doctors can prescribe specific medicines that stop bacterial ribosomes from working. So the bacteria can't make enzymes and eventually they die off. So it's one of those very unique ways that science has made connections. And that's why we have to learn about all these different interactions. So ribosomes are the machines inside your cells that are doing a lot of different and but very specific work that if you impact how they're able to do their job, build proteins, build enzymes, you're going to have significant impact. In fact, enzymes, if those are not created or if they're damaged, enzymes in your body cells are going to cause a lot of just death or something goes wrong at a catastrophic level. They tend to be very impactful. So here's an example of a CER that is connected to this concept of antibiotics and ribosomes. So the claim, antibiotics like tetracycline stop bacteria by interfering with enzymes. The evidence is these antibiotics block the ribosomes that make bacterial enzymes. Remember, we just talked about that. The reasoning is without enzymes, bacteria can't build or repair themselves, so they die. So that's a CER involving your enzymes, your ribosomes, and the application of modern medicine of a prescription for tetracycline. So that is just one example. These CERs don't need to be complicated or overly done to still be very effective at helping us contain and review our information. So hopefully we know more about ribosomes and we've made some important connections with biomolecules, with enzymes, with prokaryotic and eukaryotic, and even tying in our friend the virus.